discussions. Thank you too to our sponsors, IPNI, BASF, IPI, IFA, BBSRC, UK Research and Innovation, and Fertilisers Europe. Because of their very generous contributions, we've been able to support a lot of overseas delegates who otherwise would not have been able to come. So thank you very much to our sponsors. I unfortunately have the boring job of giving you some housekeeping notices, so please listen to those because it's important in case there are any incidents or issues. In terms of fire arrangements, there is no alarm practice expected, so if you hear an alarm, please exit by the doors at the back and the front here at each side, and we assemble opposite the doors to the conference centre on the green. If you discover a fire, please press the alarm button so that everybody is notified. Don't stop to pick up your personal belongings. Please leave as quickly as possible, and please don't try and use the lifts. If you need assistance to get out quickly, please make yourself known and we will help you. And when you reach the assembly point out there, if you can't find it, just follow one of us who know it very well, then just wait there and you'll be given instructions. And if you have any questions, please come and talk to me or any member of the staff outside who've been welcoming you. If there's a different kind of emergency, we'll be alerted, and please follow the instructions given by the staff. They'll guide you to do whatever is necessary. Please make sure that the gangways are not blocked, so we're not allowed to sit on the stairs. Please try and move any luggage out of the gangways if you can, please. But there are plenty of spare seats, so I don't think we'll be too crowded. Smoking is not permitted, but there are areas for smoking outside, so please don't smoke within the conference venue. And if you haven't turned your phone or your pager off, please at least turn it on to silent now so that we don't have phones ringing through the presentations. Now, in terms of the tours and the various trips, please, if you don't want to go on the long-term experiments tour, cross your name off. But there's a list of every delegate on the conference tour, the long-term experiments tour, and so we expect you all to want to go. If you don't want to go, please cross your name off. If you want to see the sample archive, it's really important that you sign up on one of the lists because it's very limited in terms of space. We can only accommodate 15 at a time. So please sign up, and if you haven't signed up, it's unlikely that you'll be able to go and see the archive because it's so small. Please sign up for the parallel workshops today so that we know how many people will be in each workshop, and then we can allocate the rooms. The rooms are different sizes. If it's a small workshop, we want a small room, etc. So please sign up for the workshops today. I know a lot of you have already been signing up, so thank you very much for doing that. If you have a poster, your poster board number will be identified on your badge. So please erect your poster as soon as you can this morning in the coffee break if possible, because the posters will be there to be viewed all through the 3 days of the conference. And then please, every one of us, go and look at the posters as we have coffee, lunch, tea, whatever. And there may be some other periods when we can view them too. There's a museum exhibition. We have lots of old equipment and papers, and it's very interesting to go and see some of the old equipment that was used here. Achim was responsible for helping to set that up, and we have 2 people who look after that. If you want to know where it is, ask me or one of the staff, and we'll guide you to it, and that will be open all 3 lunchtimes. And then please finally put your country of origin on the map with a sticker. Some of you have done that too, so that we know where everyone has come from. My hope for the conference is that we will all return home feeling that there is a really good and secure future for long-term experiments. Many of us have experienced the issues of funding, of the kind of question that says, haven't you finished that experiment yet? You've been going for 10 years, surely you found the results. And that's a challenge for us, even after, perhaps especially after 175 years, because politicians and policymakers just don't understand the importance of long-term experiments and continuity. So I hope that you will all go home feeling that you have global support for your long-term experiments, and when people ask you, should we continue them, you can say yes. We have international support. People feel that this is really important. And we have some questions we're going to discuss, looking at the future, and I look forward to seeing the conclusions of those at the end of these 3 days. So thank you very much, and I'll now hand over to our Director and Chief Executive, Achim Dobermann. He will give you an official scientific welcome and give the first presentation. Achim, thank you.
Thank you, Keith. I think the scientific presentations will follow after me. In my job, you have to be more of a spiritual leader nowadays, try to really encourage people to do new things. And I think that's also the general theme that I want to follow, because I believe personally that it is extremely important that we have long-term research, and as part of that also long-term research platforms or experiments. I have a personal connection to that. I think over the last 25 or 30 years I have done everything that is involved in this, creating new ones, closing old ones, changing existing ones, often against the wishes of those who had created them. I've done that at Erie, I've done that here at Rothamsted. Yeah. And I think it's part of the process that Keith has already alluded to. So we need to make sure that these things are not dinosaurs, but are actually useful and address the questions not of today, but actually of the future. So when we started thinking at Rothamsted what should be our main scientific event in our 175th year of existence, it was a pretty simple choice to say we're going to do a conference on long-term experiments. The last one we had of this kind was in 1993, when we had our 150th anniversary. So it's highly appropriate to come back to many of these things and also talk about the future. Now, of course, any talk at Rothamsted needs to start with our history, and some of my colleagues, Andy, Macdonald, and also perhaps Andy more later will come back to some of these things too, and also show more data from things that we have done. But for those of you who don't know, our founding fathers are John Bennett Lars and Henry Gilbert on the right side there. And in 1843, they basically started working together, and for several decades led the development of a fundamental understanding of plant nutrition, the role of soils and fertilizers, and all of that work was, of course, based on the money that Lars made by establishing the world's first commercial mineral fertilizer factory on a patent that he was granted in 1842 to manufacture what we would nowadays call superphosphate. Yeah. So for about 60 years, it essentially was a privately run, privately managed, privately funded research institute. It was only after that that the government played a role. I think the first time we actually received a small grant from the government was in 1911, nearly 70 years after establishing Rothamsted. We've gone through many periods of change here, so including structural changes, funding changes, and they all, of course, were dependent on how the agricultural science changed, how new innovations came along, how important agriculture was seen by government and the rest of the world. And so, but nevertheless, I think at Rothamsted, I would say, besides this tradition focus on soils and fertilizer nutrients, over the decades, we evolved quite a bit into many areas where then fundamental breakthroughs were made, led by scientists here at Rothamsted. This is by no means a complete timeline. We're actually establishing one right now in much more detail, but it just shows a few of the highlights here. In 1919, Ronald Fisher was hired to establish the statistics department. So that will be 100 years next year, another big thing to celebrate. And many of the methods that you are using nowadays, randomized complete block design, ANOVA, Fisher's F-test, all of these things came out within 10 years, essentially, of work of that group and others that followed him, like Yates. And we today still have maintained the tradition, and it also led to a spin-out company, VS&I, that commercialize the GenStat software worldwide. We also made breakthroughs in the development of a new category of insecticides, synthetic pyrethrides, which later became one of the major ingredients for pest control, but also malaria control. David Jenkinson's work and others that led to improved understanding of carbon and nitrogen cycling in the Rothamsted C model. And we just hit a milestone this week or last week. It's now being used by 3,000 people all over the world. So these are all achievements that go back to work done in the 1970s and 80s. And nowadays we've expanded, of course, and we're often in the news also for our work on GM crops, genetic engineering. And last week we planted what was probably the most complex GM trial ever conducted in the UK in terms of the numbers of different traits, and also the first genome-edited crop in the field. Actually, those are lines that come from France, where it seems to be difficult to do those trials. So it just shows you that Rothamsted nowadays is much more than what Lars and Gilbert probably even thought it would be. But I think it is also then the basis for the new strategy in science that we have established in the last 2 years, where we focus on 3 major themes or portfolios and try to tackle it within those 6 major challenges that are important for future farming, not just here in the UK, but pretty much anywhere in the world. So the first 2 focus on genetic improvement in crops, both in terms of yield, quality, resource efficiency for major crops like wheat, but also novel traits of industrial or nutritional value, like the camelina fish oil. The next 2 try to tackle this question of how are we going to do in the future intensive agriculture with less reliance and more systematic or smarter use of chemicals, because we run into resistance issues. So that requires new thinking in terms of monitoring and forecasting, but also then integration into a new generation of smarter crop protection solutions. And the last 2 challenges focus on the efficiency, performance of full agricultural systems. And that's, of course, also where long-term research, long-term experiments of the new kind play a significant role, not just here at Rothamsted, but also increasingly also in partnership with farmers in this country, and also many partners we have in other countries in the world. So we implement that through 5 strategically funded programs, 3 national capabilities, and numerous projects and partnerships, also with industry, of course. I just want to mention that we have a national responsibility that is expressed as national capabilities for long-term research. So those get funded by BBSRC on a 5-year cycle, but also receive support from the Lars Agricultural Trust here, the long-term experiments in particular. So besides the long-term experiments, you will hear a lot more about this in the next talk. I just want to mention that we also have what is probably the best in the country, if not in the world, equipped farm research platform for grazing livestock systems at Northwick in Devon, 400 kilometers away from here in the southwest, where we have 3 semi-commercial scale farmlets that allow us to test complete systems from the soil to the steak or to the lamb, and also measure everything that's along with that, including greenhouse gases and effluent into the environment. And then the third one is the Rothamsted Insect Survey, and you will also hear a number of exciting presentations with results on that. But what I want to say there is we're now going to a new stage with new equipment, new portable equipment, laser detection of insects, more molecular diagnostics to actually add more value to what used to be largely a manual process of collecting and counting and characterizing and sorting insects. So these are fantastic capabilities, and they are a clear ingredient of our science strategy. We see them really as a basic underpinning facilities, and they're open to anyone in the world. So if you have interest to work with us in these places or use data of that, contact the people in charge. It is our responsibility to enable that. So what I really want to talk in the next remaining time is three things that are often on my mind. And they pretty much represent also the culture that we have here at Rothamsted and also want to maintain or develop further. The first one is that I believe in this world where you're constantly bombarded with negative news, we need to keep long-term positive thinking alive. Otherwise, we'll all be lost. Yeah? And we lose focus on what is really important. Yeah? And that's where long-term experiments can play a huge role, I believe. Yeah? The second one is linked to that. So we need to be agile, we need to be willing to challenge ourselves, learn and change things and not just keep them as they are, also in the way we do research. And the third thing, well, that requires that we do much better, much more in how we collaborate and how we share resources, data, people, knowledge, everything. Yeah. So let's start with a question. This is the first of the 13 Gapminder questions. How many children will there be in 2100? It was a billion in 1950. It's a bit over 2 billion now. If you zoom ahead, 80 years, no, 70, or what, 80 years, yeah, will it be A, 4 billion, B, 3 billion, 2 billion? So who thinks A is the correct answer? Raise your arm. Got a few people. Who thinks B is the correct answer? 3 billion. A lot more people. Who thinks C is the correct answer? 2 billion. Very few people. The correct answer is, of course, C. Yeah. So this is how we are getting influenced by stupid perceptions and news, you know. The growth in the world population that we all worry about is largely coming from the current children becoming adults and growing older. And that'll level off, you know. But we're not really going to have a massive number of more children. You know. It is people moving up the income scale, and of course that is scary because they're going to consume more. I encourage you to go to this website. There are 13 questions. On average, chimpanzees do better in answering than the average human being. And they've tested that with groups of Nobel laureates, with groups of highly educated politicians who think they know everything, with scientists. It's always the same. Chimpanzees, by scoring average or randomly, do better than most humans because we are tempted to be concerned. Yeah. And it's something that I think is expressed then also in other data. You can see this YouGov survey from 2016, where no matter what the country was, with one exception, China, the vast majority of the people are convinced that the world is getting actually worse, you know. This survey was done shortly after the terrorist attacks in Paris, so therefore France was particularly worried, you know. Yeah. But you see how that influences people, you know. And I could debate with you for 2 hours why I think China people are more optimistic than in other countries. You know, we have a lot of Chinese colleagues here, so maybe you can ask them, you know. Yeah, yeah. But this is what we feel, you know, and it is largely coming also from this sort of display, you know, the great acceleration that's been named. You know, we see trends like this, and they are scary. They are true. They're scary. The time scale is from 1750 to 2010 here on each of those. And they show you, essentially, and that's, of course, a bit exaggerated, that the last 50 years is when everything just seems to go in the wrong direction. Population exploding, urban population exploding, we're building more dams, we're sucking out all the water, you know, people are spending all their time on their smartphones, you know, traveling too much, the carbon footprint gets up, you know, fertilizer consumption, and then the impact on the environment, on our earth systems is just negative. We're polluting, we're extracting, we're destroying the earth, and there's a lot of people who believe we're causing irreversible damage. That is probably true in some areas. Yeah? But if this leads to thinking that, oh, we're doomed, we can't do anything, well, might as well just kill ourselves now. So what I argue is we need to think about the possibilities to manage this process in the future and also put our research, our messages that come out of our science into that context, as opposed to saying everything is just getting worse. Here's another example. In 1800, 90% of the people in this world would be what we would classify nowadays living under extreme poverty. That's less than $2 a day purchasing parity power. 90%, even most of the people in this country, living in what we now perceive as misery that is largely spread in Africa. We had it here, and it took quite a while to get rid of it, So when our founding fathers started Rothamsted here, it was still 85%. But look what the progress that's been made just in the last few decades. So extreme poverty is now down to 9% of the world population. How many people of the general public know that? And see, actually, well, we've made fantastic progress. So what and you can, other things, if you are not into poverty, and like nicer things in life, you can think about, look at how many people have guitars and make nice music, hopefully, if they know how to play it, The guitar density, if you call it that way, has increased 50-fold in the last 40 or so odd years, Now, I don't know how to play a guitar, I don't have time to do it, But apparently it's an expression of people having some time to please themselves, And the money to buy a guitar, the extra money that you can spare, It's just an indicator for many things that actually have happened on a positive scale in life. For those who haven't seen it yet, there's a fantastic book by Hans, Anna, and Ola Rosling, Factfulness, just out. I've just read it. And in which he summarizes their work on the gapminder concept over the last few decades. And he basically makes it very clear that most of us are driven by 10 instincts. There's a gap between the rich and the poor. Things are only getting worse. There's only one pattern, linear change. Things are really scary. Big numbers that scare us, even though in proportion to the past or to others, they actually tend to be quite small, but we're getting scared by single big numbers. Many things are really strange. There's a destiny of the kind, like, we can't change it, we can't do anything about it, we can't fix it, therefore we're doomed. People pushing single solutions when you need to think more sophisticated, blaming each other instead of doing something about it, And there's also often an unnecessary sense of urgency that then leads to not very thought-through action. Lots of interesting examples. What Hans is arguing always over the last few decades is we need a fact-based framework to understand this world and put our actions into the right context. So, and these are some of the facts, and this is how they display it. So I think, therefore, the main role of long-term experiments, coming back to our subject, is that they are there to challenge, they are there to show what is possible, they need to be forward-looking, explain how it can be done, and why. So the mechanisms. That's, I think, the gold standard for science in that context. If we can make more use of our experiments in that context to explain what is feasible and also revolutionize the designs, the models, the methods, the questions that we're asking, then I think we'll have no problem finding the funding for it. Because people want that, people need that. People need to have more evidence to make the right decisions. And I will argue this is what we have done quite well here at Rothamsted. So you will see more of that, of course, this week, and hopefully all of you will go to see the famous Broadwalk experiment. It's, according to the Guinness Book of Records, the oldest still running scientific experiment of any kind in the world. We're very proud of that. It doesn't give us any extra money, but it's a nice thing to have. So, but when you look at that, coming back to the graph that I showed before, when it started in the 1840s, and poverty was 85% in the world, It was because yields were 1 ton. And that was just about anywhere in the world like that. And yes, it took 100 years to really get them to the levels where we are now, or more than that, 150 years. But we could see what was immediately possible with nutrients and manure. We could see what became possible once we had modern varieties of the Green Revolution kind. Capel was the first one to come in and jumped up the yields. Other technological improvements, changes in the way we manage the crop, including different fertilizer sources. Fungicides came in. We changed the fertilizer source again. Introduced new rotations. So a constant level of technological change. And this is essentially the story of modern agriculture that you can tell in just about many countries of this world, not all of them. It hasn't quite happened in Africa yet, of course. But I think what I want to argue is that if you can put the results of your long-term research into simple messages of this kind, you see no error bars in here. You see no individual years. It's 5-year averages. Who cares about this? So you can have that in the scientific papers. But I use this graph in every talk I give in the world, and I give it different meaning depending on the audience because it is relatively simple to understand. It tells a story. You can put pictures on that too if you wander around our building outside the conference center to the left and then to the right. If you spot them, there are boxes of plants hanging up there on the walls that we collected at different stages of this trial. In 1893, wheat plants looked like this. This is 70 centimeters bar here. You can see they were over 1.5 meters tall. And this is essentially a nitrogen response curve. Here is manure. Here is no fertilizer. Very clear message. 50 years later, still look pretty much the same. They had gotten a little bit shorter already because selective crossbreeding had already been invented and varieties had been developed. And then, of course, 50 years later again, we have the Green Revolution semi-dwarf varieties, and you can see the fundamental change in genetics, but we also adjusted substantially the fertilizer management of those to get the most out of these genetics. So these are pretty clear, convincing ways of displaying what we're doing and explaining this to those who believe that the world is only getting worse. So I encourage you to think of any of your experiments in that context. How can we improve the messaging of that? And how can we make changes in those experiments that are dynamic and radical when needed? We have just last year made another radical change in this experiment that Andy may mention. By bringing in a legume crop, you will see it in the field. And it's quite interesting to see, actually, at this time of the year, how that looks. And so this leads me to my second point. So we need to really be agile and be willing to make these changes. Broadwalk, when it was started, had a very simple design, just long strips. This is what it looks today. Our founding fathers may not even recognize it anymore. We have superimposed all kinds of things. I don't have time to go through the details on why we're doing this, but the primary reason is because our thinking has evolved. We have built up knowledge. We have new methods that allow us to look into new things. And we need to ask new questions with this portfolio of knowledge. And here is one. So fungicide resistance is a big topic, that affects farming right now. Many farmers are being concerned. Companies like BASF, one of our sponsors here of the conference, worry about it too because it's becoming increasingly difficult to come up with new molecules to control diseases, and the regulatory costs of those are tremendous. So if you lose effectiveness of these products in a few years because of resistance buildup, you've wasted a few $100 million. So we didn't have fungicide resistance. I'm not quite sure what the outliers. No, this year, sorry. This is the time at Rothamsted in this trial until the early 1980s. And then you can see that within 2 years. Bang. Now we can detect this because we can go back in our archive, our sample archive, and look at fungicide resistance and samples that we've kept. And we also have, when we made changes, kept a strip free of fungicides. So all the rest gets fungicides, but here we have a population that never has experienced exposure to fungicides for 175 years. So this allows us to look into those questions, and they then become part of global studies like this one just published last week in Science, showing the trajectory, the worldwide emergence of resistance to antifungal drugs in human health and food security. So Nicola Hawkins, whose work was this year before, is one of the co-authors of this study, and it's just tremendous how fast things over the last 40 or 30 years have spread, and so this is a global threat. It hasn't quite affected Africa yet because we have very little use of fungicides there, but at least in the Western world, in North America, and increasingly also in South America and in China, it is becoming a big problem. So we need long-term experiments, we need agile, changing long-term experiments to look into these kind of new questions. Another study that has inspired me a lot in the recent weeks was this paper published in Science from the Biocon experiment in Minnesota, the long-term carbon CO2 enrichment study, a phase system. Very expensive to run such a thing. The amount of CO2 that you're pumping through this, employing people to do this, the engineers, the plant scientists, the operational costs, 20 years. I don't know how much they spend on this, but millions. Most other people would have stopped after a few years because they found in the first few years the obvious, that C3 grasses responded with higher biomass to elevated CO2, whereas under ambient it was a bit less, and C4 grasses didn't show any response to elevated CO2. So most people would have probably stopped here, because it was confirming the textbook knowledge that we have about the response of plants to CO2. They continued and found a complete reversal. The C3 species didn't respond anymore in recent years, whereas the C4 species had a huge response, increased biomass through elevated CO2. It changed completely the paradigm on how we think about response to CO2, the impact of climate change on crops, maybe food production, who knows, food security. They, in this paper, found a relationship with soil nitrogen mineralization, which also reversed as a result of CO2 exposure, but only after 10 years. I don't think they can explain it yet. That's maybe a job for people like you, to think about. But unexpected discoveries of that scale, are immense in terms of importance. They change thinking, they change, in effect, possibly even in the future policies. Now, this needs to be confirmed, it needs to be understood, but 20 years is the time frame that it took to make that discovery, not 3 years of a typical project that we get funded by most of our funding agencies nowadays, with limited resources that wouldn't even allow us to build and operate a phase system. We don't have one like this in the UK for crops, at least I'm not aware of. We don't. So that's another fantastic example. Now, many countries are now entering this stage where they need to think about their future. And what you see typically emerging there is that more complex goals are become the desire of new policies, which of course means, so a lot more demand for new and more complex knowledge, and the interactions between things and the trade-offs, lots of mechanisms, so easy to get lost. We had a very interesting workshop organized by our colleagues at China Agricultural University in Beijing just a month ago, where they were presenting and discussing with experts from all over the world their concept for a green agricultural development framework in China, which is shown on the right. So 4 components: greener crop protection, better integration of plant and animal production, urban and rural development, including a village-based recycling economy, and the 4th one, new green food processing and other industries in rural areas. So build around the desire to meet these 3 major goals, and the ones in the middle as a connection. They were starting to translate this. Okay, what that mean in concrete targets, But now, of course, the question is, how can we support that with the right kind of research, long-term research, but also short-term solutions along that transformation pathway? So I think we increasingly need to think about agricultural transformation pathways, 15 years, what is it that needs to be done at different stages to get to that the desired outcome, and with the right kind of trade-off. We're having a similar discussion in the UK. I hasten to say I don't think we're as organized and planning mode as the Chinese government usually is, But, we're thinking. The government issued a 25-year environmental plan. Agriculture is kind of sort of embedded there, and in terms of things can be done. There's a consultation going on right now. It's finished. It's being synthesized on health and harmony, the future of food farming and the environment in a green Brexit. So I'm not quite sure what that green Brexit will be like, But they received 44,000 responses. 44,000. Probably a few people in this room wrote something too. But to back it up, they issued this thing here, the Future Farming and Environment Evidence Compendium, with lots of facts. This is what we are at the moment. Our productivity growth is lagging behind here and there. Our species diversity is declining, blah, blah, blah. That's where we also need to come in. Provide the right kind of evidence for these types of actions, so that in the end, whatever comes out of that becomes the right set of policies. So we're interacting with the ministry right now on this question: how do you create a workable, reasonably robust, affordable soil health management system, soil health assessment and monitoring, rewarding farmers for right things to do to your soils? So we need to make a lot of compromises in our scientific thinking because we want to make it very complicated. That doesn't work for policymakers. So we've given them last Friday 3 options. We'll see what they think about it. But that's where a lot of the evidence, our understanding, our knowledge from the long-term experiments feeds into, and the models that have been developed from those. So we just need to apply it and not just sort of get stuck in the world of scientific journals only. Those are important, but we need to do that other step. So one of our specific initiatives here at Rothamsted is for that reason. Last year, we've created a new long-term experiment. Now, I'm not prepared to say it'll last 175 years like Broadbark, but maybe, who knows? So it's much bigger than Broadbark, actually. It is much more complex. We wanted it to be relevant for what future, including more radical farming system solutions, might look like. It needed to be much more multidisciplinary from the beginning because we need to look at these synergies and trade-offs. We'd like to have it at least at a couple of sites so that you can have a generic concept but also be flexible in site-specific management. Well, in our initial stage, we say long-term, well, at least 7 years because that's what it takes to get through one rotation. But hopefully more. It needed to be reasonably interesting in terms of taking some more risks, including things that probably will fail. Has to be affordable and has to be statistically robust. And this is an interesting question here. We had a lot of arguments. What does this mean? And our statisticians have convinced us that we can do this actually with just 2 replicates. Now, most of us would have thought, well, no, that's not going to be good enough. But we think it's actually going to be good enough to answer at least the big questions that we want to answer. We may not be able to do the last bit of statistical design and analysis for every little bit, but we'll have a session on that during the conference too, and Andrew Mead is the one to ask. So this is what we are hoping to do in that space as a new activity, but essentially also create a platform I sometimes call it playground, for many scientists to work on. So anyone who has interesting questions to ask or very cool and clever new technologies or methodologies or sensors to test, come, put them in there. And I think what we also need to do in that context is increasingly work on the question of how we link our work better with the real world. We have a tendency in science to sit among ourselves, and then out there is this public who doesn't quite understand what we're up to, and why we're sitting there together and what we're scheming about, So we need to, when we design these kind of trials, but also associated satellites and activities with farmers, engage early on and co-innovate, and there will be also further talks and sessions during this conference. We are doing one thing here at Rothamsted, which we call Farm In, as an innovation network, a co-innovation network with farmers, co-sponsored by the AHTP, our commodity board here, where essentially we want to tap into farmers having a cool idea, then teaming them up with Rothamsted scientists, develop a simple scientific project without a lot of bureaucracy. We don't need to write lengthy papers for that. Test it very quickly, on farm, robustly, iteratively. And the only condition that we have is whatever comes out of that, the results need to be shared freely with any other farmer in this country or in the world. No conditions, no restrictions. And that leads me to the last bit, and that's collaboration and sharing, data. When we started at Rothamsted, these were the first data records from the first harvest of broadbark in 1844 and the first cut of the hay samples in the park rice experiment in 1856. It is very important that we keep good data. It's even more important that we process them well, that we use them well, and that we make them publicly available. So last week we had another major publication for the first time, really a systematic description of how we do this here at Rothamsted in Nature Scientific Data in our electronic archive. There will be more discussions about this during the workshop too. So we have seen an increase in external demand in particular for our data. We're not yet quite there in terms of really what should be the gold standard for open data or fair data. You will have a session on that. Findable, accessible, interoperable, reusable, fair. It's not just a matter of throwing data out there. You need to be able to use them. But it shows you the enormous effort that goes into this, here in this case at Rothamsted. It's people, it's software, it's diligence, and the willingness to make these things available early on, fast, freely. I think we can all gain much from this in science, and I think we're still too protective, too cautious, too slow in how we do this in most places. So the new long-term experiment network that we have started to initiate through a workshop last year, we'll also have a session later this week. We have started to compile data from trials that people have shared with us following a common format from all over the world. And of course, much more can be done. But you can see already the range of different crops that we are covering in this one exercise. And some initial results will be presented. So I'm hoping that this conference can really address many of these points in more detail. New questions, designs, data. Do we need living repositories, for example, for soil microbial DNA, things, indicators, linking with the real world, the global network. And of course, if anybody's got any clever ideas on how to raise more money for these things, let us know too. I've counted 37 countries participating, but please put your stickers out there that we can check whether that is correct. So with that, I think I'm really looking forward to a great conference. At Rothamsted, we actually have a partnership here. Rothamsted Research, Lars Agricultural Trust, owns all the land and buildings, and then BBSRC, which is now part of UK Research and Innovation. So we are essentially, we have a membership, we have a duty to work together to also sustain and improve the utilization of these long-term research resources. So that's our commitment. It's not easy, but we'll do it also in the future. Thank you.
Thank you, Achim. I don't think there's any time for questions, if you're happy with that. But of course, he'll be around all 3 days, so do grab him and ask a question if you've got one. Our next speaker is Dr. Andy Macdonald, who's already been introduced by Achim. Andy is the PI of our long-term experiments national capability, which you already heard a little bit about. And Andy will now tell you about some of the things we've been doing recently with the long-term experiments. Andy.
Okay. Thank you, Keith. Well, good morning, ladies and gentlemen. It's good to see so many of you here. It's my privilege this morning, as part of the proceedings, to give you an introduction to the Rothamsted long-term experiments, to put a bit more detail, perhaps, on what Achim has already said. I should say, to start off with, that just about everything I say, plus a lot more, you can find within the new revised guidebook, which several of us have spent a considerable amount of time producing, and you can find it out in reception on one of the display desks. So in my talk, I shall start off with a little bit of the history, not too much, but the history of the long-term experiments, and then go into a little more detail about the design of some of the most well-known experiments and some examples of results from early work, and also focusing a bit more on recent studies, and then maybe one or two comments about possible future uses of the long-term experiments. So the history then. As many of you already know now, that Rothamsted began as a centre for agricultural research back in the 1840s, when John Laws and Henry Gilbert established what became a series of long-term field experiments. They didn't necessarily know they were going to become experiments that would last 170 years, but that's what happened. And the focus of their work then was to look at the effects of different mineral fertilisers and organic manures on the yield of important agricultural crops at that time. So in particular, winter wheat, spring barley, long-term grassland, legumes and root crops being the main ones. And they tested the effects of fertilisers. There were new sources of fertilisers coming into use at that time. So nitrogen, phosphorus, potassium, together with sodium and magnesium, were tested. And they tested organic manure inputs as well, particularly farmyard manure from cattle, but also castor bean meal and rapeseed cake on some of the early experiments. So the initial questions about which fertilisers were important to improve production and how much you should apply were answered relatively quickly, but there were still debates about the longer-term sustainability of agriculture using mineral fertilisers. And so the experiments continued to look at those questions. And some of them continue today, and we call them the classical experiments. Perhaps the most well-known, as Achim has already mentioned, was the broad-bought wheat experiment, started in 1843 to look at the effects of fertilisers and manures on wheat production. Subsequently, it's been modified to have other crops included, so it has sections with rotations in it, and I'll mention a bit more about that later. Excuse me. The parkgrass continuous hay experiment as well, widely regarded now as the oldest continuous ecological study in the world, focused on the use of fertilisers and manures for hay production in particular. And it's come to, whilst it provided agricultural data of interest, it's now actually more ecological in that it shows us about the effects of fertilisers and manures on biodiversity of species. We have the Hoosfield spring barley experiment. Again, a little bit like broad-bought in that it has fertilisers and manures tested, but in this case on a spring crop. And we have other experiments where fertilisers were applied for some years, then stopped, and we look at the residual effects of those. We have a small experiment looking at continuous clover and the persistence of clover, and experiments that were once in arable agriculture but were then set aside and natural vegetation was allowed to regenerate, and these we call the wilderness studies. And they've been useful for ecological work to look at succession of plant communities, but also accumulation of carbon in vegetation and in soils. Some of the early experiments have been either discontinued or mothballed or modified, but they still, in many cases, provide a useful resource because the differences established within the soils from those experiments still persist, and so they can be used as resources even now for future work. Well, Laws died in 1900, but research at Rothamsted continued, and in the 1930s, other new long-term experiments were established, some here at Rothamsted and others at a smaller research farm about 40 kilometres north at Woburn, which has a different soil type. It has a sandy loam soil, also has some heavy clay soil, whereas Rothamsted, it's predominantly a flinty, silty clay loam soil. So the focus of these experiments was really to look at the effects of organic manures, things like straw, compost, farmyard manure from cattle, but also to look at the effect of introducing grass lays, relatively short-term grass lays within arable systems. But also sewage sludge was used on some of the experiments as well, the Woburn Market Garden in particular. We continue to review the experiments, and actually we discontinued some of the younger long-term experiments recently where we felt that the scientific value was limited. If we look in more detail now then at Boerbock, and you can see here the black and white photos showing how it would have looked back in the 1920s with strips of wheat running the length of a field. This is about 300 metres long, 6 metres wide. There's about 19 strips, and each having a different fertiliser or manure treatment. But in the early years, of course, we didn't have the benefits of modern agricultural machinery and agrochemicals, and so weeds became a particular problem and started to depress yields. So this led to the division of the experiment into sections so that you could fallow one section each year and cultivate it more to destroy the weeds. There were other major changes which occurred in the 1960s. It was divided into 10 sections. This was to introduce rotations on some of the sections, and as Achim has already mentioned. And in 2018, we've revised the rotation so that our current 5-course rotation on 5 sections is 2 wheats, followed by oats, wheat, and then beans. This rotation has 2 first wheats in it, so wheat after a break crop, whereas previously it only had 1. So we were looking to push the productivity of the rotation and see what effect this has in the longer term in terms of sustainability. Short wheat varieties came into use in the 1960s, and they were adopted, as you've already seen from the graph, with higher yield occurred afterwards, and this meant that we could modify the experiments to increase nitrogen rates on some of the plots. And the top nitrogen rate is now 288 kilograms of N per hectare. We have herbicides and fungicides used as they came into general use in agriculture, but we still have a section without herbicide or fungicide. And we have a section where straw has been incorporated since the 1980s. Some further modifications were made in 2001. So some of the plots we've actually split nitrogen applications to make it more consistent with commercial agriculture. Prior to that, the N was applied in a single dose in spring. So now we have plots, not all of them, but some of them that get split N applications. And plots where P has been withheld for many years, there was adequate available P, so we decided to withhold it in many of the plots, and we monitor that regularly. We also withheld S from one of the plots, sulphur, because with the decrease in sulphur deposition from the atmosphere, which has occurred in recent decades, we've expected that we'd see perhaps deficiency symptoms, so we wanted to look for that. Here you see a close-up of selected treatments with, on the far right-hand side, plots with farmyard manure, some with extra nitrogen, control plots with no inputs, P and K only on these plots, and then others with N, P and K with increasing nitrogen inputs. So you can see the lighter green or yellow colours here, symptomatic of nitrogen deficiency on the wheat there. Hoosfield spring barley, I mentioned earlier, this is another one of our important arable experiments, started in 1852. Similar in some respects to Boerbock, although it doesn't look very similar, but it does have strips of crop running the length of the field with different fertiliser inputs, so nitrogen, phosphorus, potassium, and so on. A strip with ashes might seem a bit surprising, but when fertilisers were applied, ashes were mixed with the fertilisers so that it would bulk them out so you could spread it better. So That was tested in one of the strips. Farmyard manure has been applied on various plots, one since 1852, and we've started at one more recently to see how quickly you can establish the benefits of farmyard manure on the yields of the barley. Another plot where we've introduced a higher level of P input. There are plots that have silicate at different times and since the 1860s. And the idea was to look at the effect on the strength of the straw, which was beneficial for some industries, particularly the hat-making industry, which was a local industry here in the 19th century. And P-test plots, so we have different levels of available P in the soil, and we can look at the response of the crop to P. These strips in the early years were crossed with different nitrogen inputs or organic manure inputs, so either ammonium sulfate or sodium nitrate or castor bean meal. So these are other inputs. Park grass experiment I mentioned, widely considered the oldest ecological study in the world, started in 1856. The plots run in this direction with a control plot, plots that receive farmyard manure, others that get N, P and K in different combinations. And it was noted early on that the inputs of fertilisers improved yield, as you might expect, but that it also had quite quickly an effect on species composition on the plots, with the numbers of species decreasing with the higher fertiliser inputs and grasses becoming more dominant. And further than that, those plots that received ammonium fertilisers, the species numbers declined even further over time, and it was subsequently found to be because of acidification from the ammonium inputs. So tests of lime were introduced, firstly in the early 1900s when the plots were split into two, but then subsequently in the 1960s when each plot was divided into 4 subplots, and we lime them now to maintain pHs of 7, 6, 5 and an unlimed plot. So we've got a comparison of fertilisers and lime inputs. So at harvest, of course, we take grain and straw samples, straw from selected plots. Not all of the cereal plots have straw yields done, but we certainly do grain yields. Herbage samples are collected from our grassland experiment. Park grass is harvested in the middle of June, and then a subsequent harvest in October or November. We take a small sample for yield from the herbage, but the rest is left on the plot and dried and turned as you would for hay production. And this returns the seed to the plots so that it, as you would for normal hay production purposes, it enables the continuation of the species on those plots. Grain quality is done on particularly the wheat samples of grain, so 1,000 grain weights, hectolitre weights, and Hagberg falling numbers as an indicator of grain quality. We analyse plant samples for total N and major nutrients. We do botanical surveys regularly on park grass where we take vegetation samples, separate the species by hand, dry them and weigh them. And there's been about 30 or more of these surveys done in the lifetime of the experiment. Regular soil samplings are done on the experiments. So for Bulbork, it's soil sampled. So that over a 5-year period, we've sampled all of the topsoils, and on park grass, we sample every 3 years. We measure important nutrients, total N and C, exchangeable cations, available P. This provides data for use in scientific studies, but also to help with the management of the experiment so that we can identify how much lime is needed to maintain pH and also how much P is needed to ensure that available P is not limiting on the plots where that's required. After analysing samples, we archive them. This is the Rothamsted sample archive. It actually started back in the 1840s, and Laws and Gilbert realised the importance of keeping samples. It wasn't in its current location, and it's been moved a few times since then. And you see it here in its current location. It was moved in 2009 into a refurbished facility. And there's now 300,000 samples of grain, straw, herbage, together with fertilisers and manures. And it's an extremely valuable resource because it's used not only by researchers here at Rothamsted, but throughout the world and often in conjunction with new techniques and ideas, so that you can look at questions that you couldn't have envisaged when the samples were first taken. So it's in this way that we ensure that old experiments are continuing to generate new science. And in fact, the example that Achim showed of the development of fungicide resistance has come as a result of having the archive. But it's important not only to look after your samples, but also your data, of course. And the electronic Rothamsted archive has been developed for that purpose. It contains important yields from the experiments, together with meteorological data gathered at Rothamsted, but also from the Woburn farm and from another farm at Boomsbarn to the east of Rothamsted. This data can be accessed through the data managers, and there's a lot of information on the website that gives background information about the experiments, including details of publications, of which there's more than 1,000. There's a lot of information about cropping and fertilizer inputs, case studies about how the data and samples have been used, and open access data. So these are summary data from the experiments describing important key points. It's an ongoing process, and we continue to add new data, crop yields and nutrient data to it, and develop and implement new ways of making the data discoverable and usable. So we're including DOIs now for data sets and also for publications which are more obscure and that can't be found online. You've seen this figure already. This is one of the open access data sets for Boerboek, showing yields of selected treatments, the unfertilized control, plots that get mineral fertilizers or farmyard manure. And as you've seen already, that the yields particularly have increased from the 1960s onwards with the introduction of the shorter straw varieties. But very notably, the yield potential is much higher for the first wheats in rotation. And we've seen in recent years, with the change from Hereward to Crusoe, a very sharp increase in yield. We chose Crusoe because on the recommended list, it showed higher yield potential, but still bread-making quality. And you can see a more detailed comparison here of mean yields from Hereward and from Crusoe in recent years. And you can see indeed that Crusoe has produced higher yields, particularly the first wheat in rotation, especially with the higher N inputs. So it certainly performs and with high N inputs. And in fact, we plan to change Crusoe to another variety in the autumn of this year, to Zayat, which has even higher yield potential, because we see it as one of the key focuses of this experiment to move and reflect what's happening in modern agriculture and to try and achieve high yields with bread-making quality. And you can see here a plot of grain yields versus percent N for Hereward and Crusoe. So this data would be generated over the last 20 years or so. It has both continuous wheat and first wheats in rotation. And the blue line here shows an envelope within which the data points for earlier experiments from the 1950s to the 1970s would have been contained. And you can see that the newer varieties are pushing beyond that boundary, indicating they have higher yield potential. But this is the threshold for bread-making quality, and you can see that only a relatively small proportion of the total make the bread-making quality, suggesting there's physiological constraints to achieving both high yield and bread-making quality. I mentioned that we split nitrogen applications on some of the plots, so you can see here a comparison of this is the first wheats in rotation, either single nitrogen, the green, or split nitrogen. And this is continuous wheat, either single nitrogen or split. And you can see, interestingly, that in fact there appears to be very little difference in terms of the effective yield on whether you split or apply your nitrogen as a single dose, which is a bit of a surprise. I thought that perhaps by splitting you might minimize the risk of losses and enhance the potential for uptake and increased yield, but not on this soil apparently. And there's other reports in the literature that show similar findings. Perhaps on a different soil you might find a different result. Some novel work that's been done on Boerboek. This was reported back in 2005 looking at the gene expression in the wheat plants on contrasting plots, and found that there was a high level of gene expression in the plots receiving farmyard manure compared to the mineral fertilizers, and many of these genes are associated with nitrogen metabolism. So this is something of interest at the moment. We don't fully understand the practical implications this might have, but it suggests that this sort of study might help to identify genes associated with high nitrogen use efficiency, and so may lead to conclusions that might help us with developing or identifying varieties for wheat breeding programs. Some work that Steve McGrath and Fan Giselle have been doing, so fairly preliminary studies at the moment, have been using boerbock and the Hoosfield experiment to look at how the different treatments, particularly the farmyard manure applications, are affecting the type and abundance of antibiotic resistance genes, and showing that with farmyard manure additions, we appear to be introducing new types of antibiotic resistance genes in the soil, but also increasing the abundance of several indigenous ones. So this is obviously important because of the concern of the widespread resistance genes in the environment, or increasing spread of resistance. You see here, this is another example of open access data, but this time from the Hoosfield experiment. So again, selected treatments, a little bit like the boerbock plot, showing the effect of the unfertilized control with those of receiving mineral fertilizers or farmyard manure. And again, you see a trend of increasing yields in more recent decades, with high yielding varieties coming into use. And we've been focusing more in recent years on the benefits of farmyard manure, which appear to be greater for spring crops than we found on boerbock for the winter cereals. On boerbock, you can achieve similar yields on plots with farmyard manure and mineral fertilizers. You have to add a little bit more N to the farmyard manure plots. But on Hoosfield, it seems that we can't do that. We achieve highest yields only where farmyard manure has been applied. And we think this is to do with improvements in soil structure and helping the crop to establish quickly and root well, which is perhaps more important for a crop that has a short growing period than one that's sown in the autumn, which has some time to compensate for suboptimal conditions. We established a new plot on Hoosfield in 2001 to look at the effect on yields, and you can see the pink line is the long-term manure input since farmyard since 1852, and here since 2001. And after about 10 years, the yields are not so different, suggesting that it doesn't take long to actually achieve improvements in yields with the addition of farmyard manure. Admittedly, they're quite large inputs, 35 tonnes per hectare per year on our long-term experiments. This is a bit of information I thought would be useful and interesting to you about how we're using new technologies with the long-term experiments. Our colleagues in Belgium at Leuven University came and flew their UAV over boerbock and Hoosfield, in fact, with a multispectral camera to collect the spectra from the site and when the soil was virtually bare, just a small amount of growth. And by combining this with a calibration data set, which was produced by imaging soils of known carbon concentrations at low level, you could then relate the spectra at high level, which was taken about 120 meters, with the calibration data set and produce a map. And you can see that it was able to identify the carbon concentrations in the farmyard manure plots, which are much greater than the rest of the field. So this is sort of technology that with further development might be of interest to land users and farmers to show the effect of their management on important parameters like soil organic matter. On the long-term experiments, we've seen that management affects organic carbon very substantially, with the lay arable experiment here showing that when you plough out long-term grassland, subsequently you see a decline in organic carbon when it's in arable agriculture. Similarly, when you move from arable to long-term grassland, you see an accumulation. But it can take some decades for this to become apparent, so only technologies that can help us see those changes more quickly could be useful. And I think there's scope to use long-term experiments to help with the development of these precision farming technologies, to to look at other soil parameters, perhaps phosphorus and potassium, either aerial-based or ground-based imaging. Also to look at crop mapping, because in our long-term experiments, the treatments have established differences in crop yield, so that they could be a useful test bed to develop and test this sort of technology. And in fact, we've been doing that with a group at Reading University on the park grass experiment to use low-level sensing technologies to look at biomass production and quality. But park grass, you see here another example of the open access data, but looking more here at biodiversity, really confirming what I've told you earlier, that with the addition of fertilizers and manures, the species numbers early on in the experiment, in the 1860s, decreased relative to the unfertilized control. And then with the ammonium inputs, further decreased because of acidification. And you can see here where you lime the plots, you prevent further decline in species numbers. If you use nitrate as your end source, then you see an initial decline, but it stays relatively constant thereafter. There was a decrease in species numbers on the control. We think this is down to atmospheric deposition of N and S, resulting in some acidification. You can see that where you apply lime, you prevent that further decrease. But in recent years, we've noted a decrease in the N deposition in the local area, and we've seen an upturn in the number of species, legumes in particular. There's an example of a recent study we've done in collaboration with Queen Mary University College of London and the Royal Botanical Gardens at Kew, looking at plant genome species size in the different species on contrasting plots, where they found when you look at biomass, more of the biomass is produced by species with large genomes on plots with N and P, and that when you look at species numbers, there are more species with smaller genomes on the control plot, and that when you combine this information and you get the mean biomass-weighted mean genome size, you see that it's on average greater on plots that receive N and P, suggesting the species on plots with higher levels of fertilizer are adapted better to systems with higher fertility. And this may also be indicative of, for other plants, perhaps even wheat. So it's suggesting that we need to consider genome size as a factor when thinking about how plants evolve and adapt to their environment. Here's a piece of work that was done in collaboration with the Technical University of Munich, where they used archived herbage samples from park grass and looked at carbon-13 enrichment in the herbage on plots with different inputs, so unfertilized control, plots with N, P, and K at low and high levels of N, and compared it with the change in 13C in the atmosphere. And you can see that atmospheric 13C has decreased over a 100-year period. So too is the enrichment in the herbage. The herbage is much more negative than the atmosphere because there's discrimination going on in the fixation. And if you use some convenient equations to look at the difference in the enrichment in the plants and in the atmosphere, you can relate this to water use efficiency of the species on the plots. So this is the carbon fixation per unit of water used. And when you plot this for the different treatments, you see that in all cases there appears to be an upward trend, so an increase in the efficiency of use, but more so on plots that receive fertilizers compared with the control. And we think this is a result of differences in botanical composition, and particularly that with more grasses dominant on plots that receive manures and fertilizers. So it's a sort of indication of how climate change effects may be affecting different situations in a different way. And if we think of climate change at Rothamsted, our weather data has certainly indicated that average annual temperatures are increasing over the last 20 or 30 years. And it's very likely these will be having impacts directly on plant growth. I mean, this is a sort of trend that is consistent with what we see throughout the world. But it's likely that these sorts of effects will also affect the interactions between plant growth, soils, pests, weeds, and diseases. So it could well be very complicated outcomes from climate change that may take years to become apparent. So we think it's only really by using long-term experiments and doing long-term monitoring that you'll be able to see these effects emerging and hopefully mitigate against adverse ones. We know the long-term experiments here have provided valuable information in the past to help with this, and providing we continue to maintain them and manage them well and use them with new ideas and techniques, we think that will continue to be the case for future generations. But increasingly, with discussions about the experiments here and elsewhere, it's becoming apparent that to understand the effects of climate change on global food security, then we really need to have more long-term experiments throughout the world in different climatic zones and different cropping systems, and something that's been alluded to earlier. So I'm sure that will be an area for discussion throughout the conference in the next few days. So finally, just like to acknowledge all the many colleagues, hopefully I haven't missed out too many, I apologise for those who I've missed, who've helped keep the long-term experiments going in the past and present. And thank you all for listening. Thank you.
Thank you, Andy. It's coffee time. Please put your posters up if you haven't. Please sign up for the various tours and workshops. And we come back in 30 minutes' time, just under 30 minutes. 11. Thank you.
Be good is her name. I sing my lion song, brush my mane, and she would if she could. So she pulled my lion's tail and caused me pain. She said lions are made for cages, just to look at and delight. Dare not let them walk around, 'cause they might just bite. Does she know what she does when she dances around my cage? Is her name. I trim my lion's claws and I